Anyone can tell me what are the hormones factors that are responsible for the onset of puberty? Anyone? Anyone? Because I have told you that spermatogonia will remain in dormant phase, okay, until and unless till the puberty, till the onset of puberty, isn't it? So when the person reaches at its pubertal phase, then only this spermatogonia will undergo different steps and produce the sperm, isn't it? Now there should be certain factors or hormones that will be responsible for the onset of puberty. What is that? If you look at here, the puberty starts, okay, the puberty starts at the average age of 12 to 14 years in case of male, okay, this is for the male. If you look at here, then the average can be 13 years, and when the person reaches 13 years, then there will be onset of puberty. But there is certain factors are responsible for the puberty. So what is that factor that is responsible for puberty, and that will initiates or onsets the formation of the sperm or spermatogenesis will undergo. Anyone can tell me? Please let me know. Puberty. What hormones or factor that are responsible for the onset of puberty? This question can be asked. This question has been asked in many times in your sessional examination, or in case of Viva also it can be asked. Anyone? Sunya Now, anyone can tell me? It has been taught there are puberty is there. There are different phases of puberty is there. Adrenarchy, telarchy, puberty, isn't it? There are different phases there. Now, what is that hormone? Now, that hormones that will onset the puberty. Now, if you are not knowing, then I will tell you. Basically, this is factors or hormones that is responsible for the onset of puberty is the pulsatile release of GnRH. Okay, till the person reaches pulsatile release of gonadotropin releasing hormone. So the person, when the person is in its just before the puberty, there will be no release of GnRH. Okay, there is no release of gonadotropin releasing hormone is not released. But at the onset of puberty, when there is puberty phase stage reached, then there with unknown mechanism. What is the mechanism? That still is unknown. But this pulsatile release of GnRH, is responsible for the onset of the puberty. Okay, am I clear? Which factor or hormones basically responsible for the onset of puberty? And this is also and one theory has been given. Sorry. Hello. Am I clear? Someone was there that was talking. Am I clear?
Yes, sir.
कोई बोल देना. So background but question noise आया. Other, please let me know what factor is responsible for puberty. This is the pulsatile release of GnRH, gonadotropin releasing hormone. 10 of you, please tell me yes. Then I will proceed. Otherwise, I will not proceed.
Yes, sir. Yes, sir.
5,
yes, sir.
7.
Yes, sir.
Okay.
Yes, sir.
Then I have told you that there is pulsatile release of GnRH is there, and scientists has found that basically this leptin hormone, leptin hormone is somehow responsible for the release of the pulsatile release of GnRH. Okay, leptin. That is leptin hormone is the source is adipose tissue. Leptin. The source of leptin is adipose tissue. So this shows that when the adipose tissues starts to release the leptin, then they at the onset of puberty, then there will be pulsatile release of GnRH will occur, and that will onset the puberty. Now let us discuss on other also. Okay, what is spermatogenesis? You are already knowing. The formation of sperm from spermatogonia begins with the onset of the puberty. Until and unless the person enters in pubertal phase, there will be no spermatogenesis will occur, because this spermatogonia spermatogonia will be in resting condition. Okay, that will be resting condition. They will not undergo the spermatogenesis process. The entire period of spermatogenesis from spermatogonia to this spermatozoa takes about 74 days. So the entire days for this spermatogenesis, the formation of mature sperm is 74 days. So 2 testes of human adult produce about 120 million sperm per day. Okay, 120 million sperm per day. So this can be for 74 days for the entire period of spermatogenesis. Okay, this is the thing. Now let us discuss on. Now I've told you that this one figure, this spermatogonia, until the person reaches in pubertal phase, this is in dormant phase. But when the person reaches at this pivotal phase, now these steps will occur. Now let me discuss this spermatogonia. When the person reaches at pivotal stage, then this spermatogonia starts to cell division, mitotic cell division. They will undergo mitotic cell division, and this spermatogonia, this is the divided one. Okay, this spermatogonia that will mature to form. This one is mature to form the primary spermatocytes. Look at here, primary spermatocytes. Now these primary spermatocytes will convert to secondary spermatocytes. Spermatogonium, this is spermatogonia get matured and forms primary spermatocytes, and to the secondary spermatocytes. And this process, there is cell division, meiotic division 1. So the chromosomal number.
Slide change भएन.
Okay.
हजुर सर.
अहिले देखाएको छ छैन? छ देखियो?
छैन सर.
छैन है. I'll manage. यो कस्तो प्रब्लम आइरहने छ यसो. So Acha, can you see? No? It's not visible. It's not visible. How this?
आयो सर.
Now let me discuss on. Now let me discuss on this. Okay, primary spermatocytes. I have told you that the primary spermatocyte, this is the matured spermatogonia. Then it will be formed when they are pivotal. These all the steps will be there when the person reaches at this pivotal stage. Okay, and this primary spermatocyte, this matured spermatogonia. Then this will undergo meiotic cell division 1 to form this this secondary spermatocytes. Now there is their diploid number of chromosomes 2n will be n. Okay, so this chromosomal number is reduced over here. So from spermatocytes primary to the secondary, meiotic division is there. First meiotic division. Then in this phase, the 2n will be converted into n. Again, this secondary spermatocytes again undergo meiotic division second. Okay, that will not affect the chromosomal number. The haploid and haploid. Okay, this is also haploid. This is also haploid. This is haploid. But this one is diploid. Okay, this one, and this one is diploid. Okay, and when this primary spermatocyte convert into secondary spermatocytes, at this phase, the chromosomal number is reduced. So at this phase, okay, 1, 2, 3. Okay, like that. Okay. This n. Now secondary spermatocytes will give rise to spermatids, and that will go on differentiation. That process is known as the spermatic conversion, spermatid to mature spermatogonia. That is differentiation or final maturation. It's known as spermiogenesis. Okay, This is also the part of spermatogenesis. Now if you look at here again, now if you look at it now, the slideshow also I will show. If you look at a one, okay, this one spermatogonia will produce, okay, about 512 spermatids, mature sperm. Okay, about they will produce 512 mature spermatid or mature sperm. One spermatogonia. Now spermatogonia proliferate mitochondrial division with the onset of puberty, that I have told you. When there is onset of puberty, this spermatogonium proliferate, or they will start to produce different their daughter cells, or like that they will form the different number of cells and mitotic division. Now mature spermatogonia are called primary. Primary undergo meiotic division, reducing the number of chromosomes, and that secondary spermatids and spermatozoa have haploid number of chromosomes. Okay, this is the thing that is important, so I have highlighted on this one. Okay, this primary to secondary reduced, have reducing number. Now if you look at here, now conversion of mature sperm. So the conversion of these spermatids into mature sperm is known as spermiogenesis. Spermiogenesis means conversion of the spermatids to mature sperm. This is known as myogenesis, and it takes about 21 days. It takes about 21 days under the influence of hormones like FSH and estrogen that I have discussed. Recently, this is the importance of the estrogen. So the testosterone are required in all the phases, but estrogen, if you look at here, estrogen, this has got main important role on this phase. Estrogen. So estrogen is main important. I have told you estrogen also produced from the Sertoli cell, isn't it? They are also producing the estrogen. Now if you look at here, the hormonal factor that influence spermatogenesis, what are different hormones that will influence the hormone that will stimulate the spermatogenesis formation of sperms are testosterone, LH, FSH, estrogen, and growth hormone. Okay, growth hormone. Why the growth hormone? Suppose the person is having pituitary draft. Okay, pituitary draft. In case of pituitary draft, growth hormone is reduced. Then it can lead to the infertility also. Okay, so draft vision can lead to the pituitary draft. Okay, not other draft. The pituitary draft can be infertile due to the absence of the this growth hormone. So growth hormone is important for the this is the important function, and there are different hormones that are also responsible for spermatogenesis, and they will influence the spermatogenesis. Clear? Am I clear? It is clear.
Yes, sir.
This portion is clear, isn't it? Now let me discuss on this one. Okay, semen ejaculated during each ejaculation average about, so from each ejaculation the semen that will be released is about 3.5 ml of semen is released per ejaculation, and sperm count is about 120 million per ml of semen. So there is 3.5 ml of semen is there, you can multiply, and that amount of the sperm will be released, okay, for ejaculation. But if question is asked what is the sperm count, then you have to tell that it's about 120 million per ml of semen. If you look at here, my previous slide also, I have told you that look at two testes also produces about 120 million sperm per day. Okay, 120 million sperm per day will be produced, and if you look at here also 120 million sperm per ml of semen. Okay, this can be taken for your easy. You can correlate on that also. So normal sperm moves in fluid medium at velocity of, so at the velocity of 1 to 4 mm per minute. This is the normal velocity of the sperm, and in some books they have written that about the average velocity is about 3 mm per minute. So this is the velocity. Now if you look at here, sperm life, what is the sperm life? How much this ejaculated, okay, sperm will survive in the female genital tract? It's about 1 to 2 days. It will only survive for 1 to 2 days in female genital tract. But if you want to store the sperm for years also, you can store. The cryopreservation or different preservative will be there and they are using. But in relation to female genital tract, the life is about only 1 to 2 days. Anyone who can tell me the average life of the ova or the ovum? Anyone who can tell me in the female genital tract?
24,
Sorry?
24 to 48 hours.
24 to
48 hours.
Okay. 24 to 48 hours. कहाँ पायो तिमीले त्यो? हेलो को बोल्यो भन न from which
Google search.
हजुर
Google search गरेको.
गुगल सर्च भन्ने हुँदैन नि बुक पढ्नु पर्यो नि है
हुन्छ सर.
So it's about 2 to 3 days, okay? 2 to 3 days. The life expectancy of ovum or ova is about 2 to 3 days. For the sperm, it is only 1 to 2 days. Okay. Now let us discuss what the capacitation of sperm. Now I have told you, just if you look at here, I have told you that how this sperm is being formed and what are the in which stage the sperm will be formed, or beginning of the sperm, production of sperm at the onset of puberty, the sperm starts to form. And I have told you the time required and how much it will produce and how much quantity it will produce and different steps for the formation of this sperm, isn't it? Now the sperm is being formed. Now suppose it has been ejaculated. It's in the female genital tract or in the vagina or cervix. Then so different mechanism will occur. Okay, again then that is known as capacitation of sperm. What is that capacitation of sperm? That capacitation of the sperm will be occurring in female genital tract. Okay, female genital tract, this capacitation of the spermatozoa will occur. It's not in the male reproductive tract, but it's on the female reproductive tracts that capacitation of the sperm will occur. This is the final maturation. This is the final maturation of the sperm or spermatozoa. The final maturation of the sperm will occur in the female genital tract. So this capacitation will make the sperm able to fertilize the ovum. Okay, this is the final preparatory physiological changes. This capacitation of sperm is the final physiological changes that occurs in the female reproductive tract with and that results the sperm able to fertilize. So it will make the sperm viable, competent to fertilize. So what is that capacitation of sperm? What is that final physiological changes? What is that final maturation development process of this spermatozoa? That is known as the capacitation of sperm, and it involves 2 stages. Basically, it has got 2 stages. One is increasing the motility of spermatozoa. So there should be increase in the motility of spermatozoa. Suppose the sperm is deposited. In female reproductive tract, then there should be increase in the motility of sperm or spermatozoa must occur. Otherwise, the fertilization will not occur. And another is facilitating their preparation for acrosomal reaction. So another is also during the capacitation of the sperm, 2 things will happen. One is motility of the sperm or spermatozoa increases in tremendous way. So motility of the spermatozoa increases. Another is that it also prepares the sperm for acrosomal reaction. It's preparation only. Okay, acrosomal reaction actually occurs in one of the steps of the fertilization, but it also facilitating their preparation for acrosomal reaction. So in capacitation, one is occur this one. Another is that preparation for the this acrosomal reaction. But now question is how they will occur. Anyone who can tell me? Anyone who can tell me about how this motility of spermatozoa once get inside this female reproductive tract get activated? Anyone can tell me?
Prostate specific antigen hydrolyzes the seminogenin of semen.
Okay. This can be the one, isn't it? And this can be one that I have told you in your previous class also. Prostate specific antigen is there, and they will coagulated. One will become liquefied, and that will releases, or that will free the sperm to get motile, isn't it? Another is also there. This is main important one. Okay, this seminogenin. Okay, this is also one of the factor. Another factor, main important factor. This is the loss of decapacitation factors. Okay, this uterine and fallopian tube wash away various inhibitor factors that just you have told me about this seminogelins, isn't it? One of that can be seminogelins, or other factors is also there, but precisely what factors is there that has not been pinpointed. But I have told you that this prostate specific antigen, they are produced from the prostate, isn't it? But the capacitation of sperm, that will occurring in the female reproductive tract. So female reproductive tract, in relation to female reproductive tract, we have to tell about the capacitation of the sperm. Let's suppose, so in relation to the female, this uterine and fallopian tube, there are fluids are there, they will wash away these inhibitor factors. Okay, there are different inhibitor factors which comes along with the semen, that will be washed away. Okay, washed away by this uterine, fallopian and fluid. Okay, now another main important one. Okay, this is another main important, increasing the motility of this spermatozoa in the female genital tract. One is this this loss of the decapitation factors. Loss of the decapitation factors, I have told you, different decapitating factors are being present in the semen. So this has to be removed, and they are washed by the uterine and the fallopian tubes fluids. Now another is, there is also increased activity of the sperm specific calcium channel, cat spore. Okay, so increased activity of this cat spore. So there is, this is cat spore, this is the channel. Okay, this is the protein, and this is alkaline sensitive calcium channel. Okay, alkaline sensitive calcium channel, they are actually present on the principal tail of the sperm. So at the principal tail of the sperm, this cat spore protein is there, and this is the calcium channel. This is sperm specific calcium channel. Calcium, this is calcium channel, sperm specific. So this name is sperm specific calcium channel. So the activity of this cat spore, look at here, cat spore, this is the membrane, this is the principal tail. Okay, one portion of the principal tail of the sperm, and the influx, there will be influx of the calcium. There will influx of the calcium from the cat spore. This is surrounding, and this is, if you look at here, there is the opening of this calcium channel. So calcium will be influx from the cat spore, this calcium channel. Okay, so when this sperm, what makes this cat spore or this calcium channel will be activated? What makes? One is that one factor that will activate this cat spore calcium channel is movement of the sperm from acidic vagina to the alkaline cervical mucus. So when the sperm moves from acidic vagina to the alkaline cervical mucus, okay, the pH will be from acidic to alkaline. So this alkaline, this alkaline nature of this mucus, cervical mucus, causes the activation of this cat spore calcium channel. And when this cat spore calcium channel get activated due to the movement of sperm to the cervix, and this cat spore protein or calcium channel is present in the principal tail of the sperm, so alkaline medium causes the activation of this cat spore calcium channel, and this calcium will get influx. When the calcium get influx, you are already knowing calcium, then the motility of the sperm, this flagellar motility will be tremendously increased. This is known as hyperflagellation. So previously the movement of the sperm was quick undulating motion. Now that will convert it into the powerful whip-slaps motion. So their motility of the sperm will be increases due to the influx of the calcium from this calcium channel when the sperm reaches to the alkaline medium. Am I clear?
Yes, sir.
Arlin Bhana, am I clear? Please tell me, other. Please tell me. It's clear or not?
Yes,
It's important.
Yes, sir.
Yes, sir.
Good. This capacitation of this spermatozoa or sperm, mature sperm is important for examination point of view. Okay, capacitation of sperm, and this is in a university exam. So you should be able to write on this thing. Okay. Another factor that also increases the motility of the sperm. Another factor that also increases the motility of this spermatozoa is the sperm moves away from this cholesterol vesicles. The cholesterol vesicles is also present in semen. So when after ejaculation, the sperm will move away from these vesicles. Okay, that enabling sperm and that will enables the sperm for this fertilization. Okay, and that enabling acrosome much weaker. So this also facilitating the acrosomal reaction. This is facilitating only. Okay, preparing this one. Okay, I have told you this one, isn't it? This one. Okay, facilitating their preparation for acrosomal reaction. So the removal of the cholesterol from, or this moving away from the cholesterol vesicles, the sperm moving away or moving away from this cholesterol vesicles will facilitate their preparation for the acrosomal reaction, because it will make the acrosome weaker. So this cholesterol is covering the acrosome. Okay, the cholesterol is covering the acrosome. So when the sperm moves away from this cholesterol vesicles, then what will happen? The acrosome that will become the weaker, isn't it? So it also facilitates for the acrosomal reaction. Now there are different stages there in fertilization that I will not discuss right now in mere detail about fertilization, the steps in the fertilization, but for the fertilization I'll discuss only few concept regarding this fertilization. This fertilization occurs in ampulla of this uterine tube or fallopian tube, ampulla, and the ampulla of this uterine tube or fallopian tube, the fertilization will occur. This question can be asked in the MCQ like that. Okay, but the site of fertilization is ampulla of the fallopian tube. This can be the one question. And now if you look at here, this is the stage. Okay, that will show the steps or sequential event in the fertilization. If you look at here, the sperm is there. This is capacitated. Okay, this is capacitated. The sperm is already being capacitated. The capacitation of spermatozoa is already gone there. So this capacitated sperm, then they will have this powerful movement is there, with slap movement is there. They will reach to the ova. This is the section from the ova. Okay, this section from the ovum. Okay, from the ovum. If you in the ovum, you can see there is granulosa cell is there. Okay, there granulosa cell is there. Remnant, some remnant of the granulosa cell is there, and there is zona pellucida is there, and other membrane is also there in the ovum. So when the capacitated sperm reaches to the ovum, then if you look at here, the acrosome is there. Okay, this acrosome is there. Acrosome is there. This acrosome consists of the hydrolase enzyme. Okay, this consists of this hydrolase enzyme. Okay, am I clear? Is this clear? Hyaluronidase enzyme, okay? Sorry. This acronym consists of hyaluronidase enzyme, okay? Hyaluronidase enzyme. This acronym. This keep this the cap, okay? This is the head, and the cap. Acronym also consists of the hyaluronidase enzyme, and that hyaluronidase enzymes will dissolve, okay? Dissolve the granulosa cells, some of the remnant or granulosa cell. They get this granulosa cell. They act this granular cells, then they will dissolve by this enzyme present in the acrosome, hyaluronidase enzyme. Now the sperm reaches to the zona pellucida. Now sperm reaches to the zona pellucida, and it will bind to the JP3 receptor. So they will act. They will bind on JP3 receptor, and there is release of. There is again, if you look at here, then binding to this zona pellucida. When this, then acrosomal reaction occurs over here. Then acrosomal reaction occurs, binding of sperm with the zona pellucida. Then acrosomal reaction will occur, and that will release the acrosomal enzyme. So when this sperm binds to zona pellucida, there is release of the acrosomal enzyme. Okay, acrosomal enzyme, that is mainly the acrosin, and that will help to penetrate. Okay, that will help to penetrate. And if you look at here, this is penetrating. Okay, this is penetrating, and ultimately it reaches to this portion membrane. Okay, this is vitelline membrane, like that. Then there will be the only the of this one nucleus. Okay, pronucleus. This pronucleus will go inside this, and this male and female pronucleus get combined, and they will form the zygote. Am I clear?
Yes, sir.
Any confusion? So this is the step, okay? The chemical substance will be produced by this ova, and this also facilitates the movement of the sperm as well, okay? So the olfactory, so olfaction, from the olfaction also the sperm can move towards the ovum, one of the factor, okay? Then it reaches to the then they will hyaluronidase. I have told you that hyaluronidase enzyme, okay? Then they will dissolve the granulosa cells, and they will bind to the zona pellucida, JP3, okay, JP3, and ultimately they will penetrate acrosin enzyme. This is the acrosin enzyme, very important enzyme, and ultimately it will reaches to the ovum, and the nucleus will go inside. Only the nucleus of the sperm will enter inside, and pronucleus of male. This pronucleus is the pronucleus, okay, of sperm, and the pronucleus of ovum is there. They will fused. Am I clear? Am I clear?
Yes, sir.
So this is the thing that you should be knowing, okay? Acrosomal reaction. Where the acrosomal reaction occurs? Acrosomal reaction actually occurs at this stage, okay? When the sperm reaches to zona pellucida, then there will release of acrosin from the acrosome, and that causes the penetration of the sperm and reaches to the inner or cytoplasm of the ovum. Only this male pronucleus will reach, other will be there. Now another question can be, so this is the thing that is addressed zona pellucida and memory structure surrounding. This is the memory structure around the ovum, and acrosin reaction and penetration of zona pellucida. But this is, if you look at here, addressed from here to the cell membrane of the ovum with the break of fusion and release of nucleus into the cytoplasm of the ovum. This is the thing that this is the step. Now another, and now one of the question that is most important. One of the question is, okay, I will write it here. What is that question that I will be writing? Why does only one sperm enter the OC by TT. This is the question. Why does only one sperm enter the oocytes or ovum, not other one? Other do not get enter, isn't it? Their polyspermy is being prevented. This is the question is indirectly saying that how this polyspermy get prevented. Anyone can tell me why only one sperm enters the oocytes? Anyone can tell me? Anyone can tell me? Ajit?
When one sperm penetrates the ovum, and there is certain membrane change occur in the oocyte so that another sperm cannot penetrate.
Okay, that membrane potential will be changed, isn't it? One of the factors is the membrane potential will be changed. That can be the one answer. Other? Anyone? Another? Anyone can tell me? So please try to find this answer, okay? Please try to find the answer of this question. One is I will slightly give you hints on this. So there will be JP3 block will be there, okay? Another is cortical reaction. One is it is related to cortical reaction. Another is JP3 block. What is JP3? This is actually present in the zona pellucida. Okay, this is one of the receptors. I have told you that when the sperm will bind on JP3 receptor, only on that condition, there will be acrosomal reaction will occur, isn't it? So when this sperm bind with JP3 receptor of this zona pellucida that is present on sperm, the acrosome reaction will occur, and that acrosome when released, then it will be able to penetrate, and this pronucleus, male pronucleus, will be power or reach to the cytoplasm. Now my question is, now my answer will be, it is related to cortical reaction, okay, cortical granule reaction, okay, and another is JP3 block. What is JP3 block? And cortical degranulation. What is that? Okay, or cortical granules, that will be exocytose. This cortical granules actually present on this ovum. Okay, this cortical granules, let's get exocytosed. Okay, and certain thing will be occurring, and JP3 block will be there, and that will causes the blocks the polyspermy, and other sperm cannot get inside this ovum. So I have given you a clue on this, isn't it? Cortical granules from this ovum get exocytosed due to one mechanism, and that will causes the JP3 block. And JP3 block, if it is there, JP3 block is there, then no more sperm cannot bind with the JP3 receptor, isn't it? JP3 block is there, then sperm cannot bind with the other sperm cannot bind with the receptor JP3, and this acrosomal reaction will occur, and no entry of this or this pronucleus, male pronucleus, to the cytoplasm of the ovum. Okay, okay. If you want, am I clear? Am I clear?
Yes, sir.
बुझे ना बुझे है. Cortical granules from the ovum get exocytosed, and that will causes the JP3 block. And JP3 block, if there no furthermore binding of the sperm with this JP3, and no acrosomal reaction and other process as well. Am I clear?
Yes, sir.
Don't find out, okay?